Greetings students, welcome to today's session that is focusing on how do we figure out the amino acid sequence that forms a particular protein. Okay. Proteins are such important players within a cell, all the central dogma from DNA to RNA to proteins all end up in proteins, and then the proteins form the majority of functions within a cell, right. And so, it is important for us to understand what is the sequence of amino acids that make up a particular protein, and we are going to look at it by understanding the spectrum of a protein. We will shortly define what a spectrum of a protein is. Each protein has a unique signature called a spectrum, and that spectrum will be used to reverse engineer or to reconstruct what is the original amino acid sequence. So, in that sense, this is a reconstruction or a realization algorithm that we are going to look at. Okay. Similar to the other topics that we have dealt with, this will also follow a similar style, but on this very important player, basically the protein or a peptide. A peptide, you can think about it as a small protein. So, it is a small protein sequence. So, that is a peptide. Okay. So, we want to figure out the ordering of amino acids, the sequence of amino acids that make up a particular protein or a peptide. Okay. And then how do we figure out the sequence? From some measurements, experimental measurements that will give you the spectra of a particular protein or a peptide. What do we mean by a spectra? A spectra is in very simple terms. Okay, in actual reality it is a bit more complicated, but we will simplify it and say that a spectra is the masses of all the substrings of a particular peptide. Okay. We will clarify what these masses mean, but there are certain instruments called mass spectrometry that will give you some idea about the masses of the different subpeptides, that is, or the substrings of a longer peptide. Okay. Now, given these masses of this different substrings of a peptide, and then given the mass of the total peptide, you want to figure out what are all the amino acids and in what order these amino acids are present within that peptide. Okay. And that is the computational problem. You want to reconstruct a peptide sequence from the masses of all its substrings. Okay. We are going to look at brute force algorithms for it, exhaustive or enumerative type of algorithms. It may be a little bit of branch and bound, okay, so that we are able to figure out the peptide whose ideal spectrum, right. If you know the sequence, it will have a particular ideal spectrum that matches the observed spectrum from the experiments. Okay. And as I have said before, proteins are very important players, and it is important to understand the sequence of amino acids mainly because of the relation between sequence, structure and function. This was true in DNA as well. Knowing that the sequence of DNA told something about what the function of the DNA is. In proteins, it is even more important knowing the sequence of amino acids, which is shown here in these little beads, right. Each bead here is an amino acid and they are all stringed together into this long peptide sequence, and these sequences then form into secondary and tertiary structures, and once they fold into these nice structures, then it goes ahead and performs the function. So, there is an intricate relation between sequence and structure and function. So, knowing the sequence will tell us a lot about what the structure and the function of the protein is. That is one major use, and these functions could be like what other proteins that this protein interacts with, or other drug molecules that this protein interacts with, right. And then the evolution of this protein within a family of related protein sequences, whether this protein stays in one cell compartment or does it shuttle or transport to other compartments. So, there are certain small peptide sequences called signal peptides that are attached to the end of these proteins, and that is kind of like a barcode or a zip code that will tell where this protein should be shipped to, right, within a cell. So, understanding the amino acid sequence, the peptide sequence will tell us information about all of these things, either using computational means or experimental means. Okay. Computational or experimental methods that try to understand the structure and function of the proteins can greatly benefit from knowing the sequence of amino acids in the protein. Okay. Now, this is not only to understand the sequence, structure, function relationships, it is also used to design experiments that will quantitate these proteins. Okay. There are certain experiments where if you know the sequence of a protein, you can then better design that experiment. Aptamer based design is a modern technology for, or a more recent technique for understanding the concentrations of proteins, right, or the abundance levels of proteins in a particular sample. Okay. In a cancer sample, for instance, compared to a normal sample, this can be used to figure out biomarkers for cancer, protein biomarkers for cancer. So, in order to quantify the levels of protein, you can use certain experiments which can benefit the design of those experiments, and these experiments can be optimized by knowing about the sequence of the proteins that you are trying to measure. So, it is important to know what the sequence of a protein or a small protein called peptide is, and we are going to look at it in the context of a particular antibiotic which is a small peptide, okay. And that is the exposition given in the book, and we are going to follow that along to understand how to figure out the sequence of amino acids that form or that constitute this particular peptide, an antibiotic peptide. And before we delve into the sequencing problem, let us just briefly look at what an antibiotic is. You may have heard about penicillin as an antibiotic that was discovered by Alexander Fleming, and there are other naturally occurring antibiotics as well, right. And then these are basically substances that kills a bacteria, okay. And this is occurring naturally because of evolutionary warfare, arms race between the fungi and the bacteria that try to feast on a particular nutrient, okay. We will particularly study a small protein, small peptide called tyrocidine B1, okay. The sequence of amino acids in this peptide is given below. So, there is a valine followed by a lysine, leucine, phenylalanine and so on. So, you have VKLF, these are all symbols representing the amino acid. So, we have VKLF, P, W, F and Q, Y, okay. So, that is the sequence of amino acids that form up this particular short peptide, this antibiotic called tyrocidine B1 produced by Bacillus brevis. So, the problem we have at hand is that we have a particular protein or peptide that we want to sequence, in this particular case tyrocidine B1, and we want to know the amino acids that actually the content of the amino acid, the composition of the amino acids and also the ordering in which the amino acids are present in the particular peptide, okay. So, from these question marks we want to be able to go to this ordering of amino acids, okay. And before we actually go ahead and address the sequencing problem, let us just look at how this protein may be produced, okay. Maybe that will give us some clues into how we should approach this problem, okay. So, how do bacteria make antibiotics, or how are proteins produced in general? We know very well that the central dogma of molecular biology, that DNA makes RNA and an RNA makes protein, right, in colloquial terms. In formal terms, DNA is transcribed into RNA, right, where you have all these letters, these 4 bases ACGT in DNA that gets into these counterpart letters ACGU in RNA, right, where a T is replaced with a U. And then from the mRNA, then you have a genetic code that will convert every triplet or a codon into a particular amino acid, right. So, that is shown over here. You have ACGU and then all possible triplets or these codons that are made out of ACGU. It is 64 3mers in number, 4 times 4 times 4, right, and then that is getting mapped into 20 amino acid using the genetic code, right. And for instance, if you look at an example of UGG, that gets mapped to tryptophan or W, and then because there is 64 3mers or 64 triplets or 64 codons and only 20 plus amino acids, right, we can have redundancy in the genetic code. So, multiple codons or multiple triplets here CUA, CUC and so on can get mapped to the same amino acid lysine here LYS, or indicated as a symbol L, right. And then of course, there are certain triplets which code for stopping the translation process, okay. So, these are the stop codons, okay. Now, we may be interested in figuring out which part of the Bacillus brevis genome, right, which gene in the Bacillus brevis genome encodes for the tyrocidine B1, okay, because the central dogma says that proteins all come from DNA via the RNA intermediate, okay. So, we are interested in finding out which part of the genome, which gene in the genome of Bacillus brevis codes for this tyrocidine B1 protein, okay. And let us say that some oracle has already given us the sequence of amino acids in the tyrocidine B1 protein. So, they have told us it is of length 10 and then these are the first, second, third and so on until the 10th amino acid, okay. And the goal is to figure out which 30mer, because each amino acid is coded by a codon of length 3, right, a triplet or a 3mer. We are looking at a 30mer in the genome of Bacillus brevis that will code for tyrocidine B1, okay. And because of redundancy in the genetic code, there are many different 30mers that could code for the same protein tyrocidine B1, and because the translation can happen in any of the starting positions, right, you have to slide a window of length 30 across the whole genome, okay. So, you can't just look at multiples of 3, but you will have to slide a window one position at a time and slide a 30mer window and then see if that 30mer, once you apply the genetic code mapping, will give you this tyrocidine B1, okay. Once you do that, you know what happens, something surprising happens. you just look for a 30mer in the genome that will code for this tyrocidine B1, and then you keep searching for it, you keep sliding all these windows, right, and then you keep searching for tyrocidine B1. And also there is one other detail that I missed out. So, tyrocidine B1 is a circular peptide, right, it is a cyclic peptide, and so you look at different linear representations of this cyclic peptide, you are looking for all of those linear representations in the genome, and then you never find it in the genome, right. And that is pretty interesting, because it is saying that somehow the central dogma is wrong. I mean, we know that exceptions can happen to central dogma. So, then what is really producing tyrocidine B1? Okay. This is kind of a computational way of verifying that none of the 30mers in the genome are actually coding for any linear representations of tyrocidine B1. And what do I mean by a linear representation of tyrocidine B1? So, you have a circular peptide and there are 10 different linear representations. You can break it here, you can break it here, you can break it here, and so on, right, and then make it into a linear representation. So, none of those linear representations are encoded in the Bacillus brevis genome. Okay. So, what could really be happening? Experimentally also this was verified that the tyrocidine B1 was dodging the dogma, it was an exception to the central dogma, right. That in 1963 what Edward Tatum did was inhibit the ribosomes that do the translation process, right, and then looked at all the proteins that are still produced, right. If you inhibit the ribosome, if all the proteins are produced by the central dogma, then no other protein should be produced, right, but then some peptides were still produced, including tyrosidines, okay. And so this means that there is some other mechanism outside of the central dogma where the proteins are produced, okay, and through which the proteins are produced. So, we cannot just look at the DNA sequence alone and the RNA intermediary and the genetic code to get some ideas about what the sequence of a protein could be. We need to directly work with proteins as a prime player, right, and then try to look at its composition and then sequence directly from working with the protein molecules. And in this particular case, there was this set of peptides called non-ribosomal peptides. These are peptides that are produced outside of the central dogma, okay, and then including the tyrosidines. And this is produced by another protein complex called NRP synthetase, which adds one amino acid at a time, again not encoded in the genome at all. So, what we have is a computational and an experimental support that this tyrocidine B1 is not encoded in the genome and that it is produced through some other means other than the central dogma. Okay. And what people have figured out, researchers have figured out, is that this is through a mechanism called NRPs, non-ribosomal peptides, okay, and there is a particular complex called NRP synthetase which produces such non-ribosomal proteins outside of the central dogma. It does not require the encoding of this protein in some gene in the genome.